Hello again, biologists. This is Mr. Kinnear with a video lesson for higher biology. This is the first of our video lessons for Unit 1, DNA and the Genome, and we're looking at Key Area 1.1, Structure and Organisation of DNA. So this is what DNA and the genome looks like. We've got 8 key areas that we're working through in various video lessons. We're starting at Structure and Organisation of DNA. Now, many of the key areas and the learning that we're going to be doing is building upon previous knowledge and information that you've picked up in S3 and National 5 biology. But obviously, because we're at higher, then we'll be adding an extra layer of detail to that as we go through. So as I said, you will know some of this information from previous biology lessons. I'll show you this picture of the Cayenne Tower in Dubai, which might have a familiar look to it, because it is actually inspired by the structure of DNA, the double helix. So that twist is to mimic the idea of the structure of DNA. So what I'd like you to do is have a quick think about what you have learned so far about DNA. So your S3 biology or National 5 biology, what do you know? So even just take a minute to pause this video and write down a few bits and pieces that you remember, because we'll build upon that in the coming information.
So hopefully you did get a few bits and pieces written down there. We're going to look at the key concepts, and we're going to look at building, as I said, an extra layer of detail in higher biology to what you previously know. So you should know that all cells store their genetic information in the base sequence of DNA. And also that word genotype is something that came across in National 5 last year. The genotype is determined by the sequence of bases. So hopefully part of what you did know, and you wrote that down earlier, was DNA. What does it stand for? Those 3 letters. What does it stand for? Hopefully you're all shouting at the screen now. Deoxyribonucleic acid. Okay. You should also know that the DNA molecule is comprised of 2 chains of nucleotides. That's something we learned in National 5. And this is a diagram that should be fairly familiar to you, with the hydrogen bonds down the middle, the sugar phosphate down the 2 sides. The nucleotides, the most basic unit of DNA, are something that should be familiar to you. We drew them quite extensively in National 5 biology last year. So they are made up of 3 units, a phosphate, deoxyribose sugar, and a base. One new bit of information, however, is that they have different ends, a 5' end and a 3' end. So that 5' and 3' are pronounced 5' and 3'. And we'll come back to those in a couple of slides going on. But you should remember that those bases can be one of 4 different bases. So there are 4 different types of nucleotide, and that is down to the base that is attached to the deoxyribose sugar. So the 4 bases in DNA: adenine, thymine, guanine, and cytosine. Last year in National 5, you might have got away with just knowing ATGC. However, in this year, you definitely need to know at higher level adenine, thymine, guanine, and cytosine. And the big question, why is that important? How do these bases pair up? The simple one, I remember it, why, is apples on trees, cars in garages. And as you can see, A and T go together. They have 2 bonds between them, and guanine and cytosine have 3 bonds in between them. So A and T always go together, G and C always go together and never mix up. These then are known as complementary bonding. So that base pairing is complementary. Whenever you see an A on one side, you get a T on the other. Whenever you see a C on one side, you get a G on the other. And that is a complementary base pairing. And you should get a hint there what type of bond is holding those bases together. So even if you don't remember, have a look at the diagram and have a little think. It is, of course, hydrogen bonds that go between those base pairs. So as I mentioned, when we're talking about the nucleotides, we said that there was a 5' end and a 3' end. But what on earth does that mean? Well, let's have a closer look at the sugar to think about numbering the carbon. So as you can see, we've got a ring, a pentagon shape, is a ring, and it's a ring of carbons with an oxygen molecule at the top. Now, it might look scary, particularly if you're not doing chemistry, but we'll simplify it a little bit more. So we're going to start here, C1, C2, C3, C4, and C5. So obviously that then gives us a bit of an indication of 3' and 5'. But let's simplify it a little bit more. Okay, so this ring, taking a bit of information out, but again, same thing. Carbon 1, 2, 3, 4, 5. And again, our 3' down here, and our 5' up here. And the 5 and 3' ends are important when it comes to adding more nucleotides into the chain of a single strand of DNA. So the 5' end joins at the phosphate group, the 3' end joins at the deoxyribose sugar. And to add another nucleotide in, it's extended or grown by adding nucleotides to the 3' end. So as we can see here, we're adding a new nucleotide at the 3' end, which joins to the 5'. We still have then a free 3' end at the bottom there. That is ready then to join to the next nucleotide that's going to come up and join again to create a third nucleotide in our strand. And this term should be familiar to you from last year. When these nucleotides join together, they join together via covalent bonds, and that then forms the sugar-phosphate backbone. It then forms a polynucleotide chain. So poly meaning many. So the units of nucleotides join together, many nucleotides or polynucleotide chain will form one single strand of DNA. And we know from last year that it's not just one single strand of DNA. DNA is double-stranded. However, something new for you this year is that each strand runs in opposite directions from one another. This allows complementary base pairs to match up, and it's also known as antiparallel. Okay, so can you just think for a second what an antiparallel sequence might look like? And as you can see, we've got 5 to 3, 5 to 3 on our left-hand side. So what does an antiparallel sequence look like? Well, we start obviously slightly opposite in that we have 3' at the top going down to 5'. So on left-hand side, 5' going to 3'. On the right-hand side, 3' going to 5'. So that's a new bit of information, this antiparallel structure. You can see that the sugars are pointing in different directions. You can see that the phosphates are pointing in different directions. Your bases in the middle, there's probably not much difference from your previous learnings about DNA, but the point of the sugar definitely going in different directions, and the phosphates coming off them definitely going in different directions. So that creates our antiparallel structure. So they're both in parallel lines, they're joining up at the bases as before, but this time our sugar and our phosphate are going in different directions. And that then again is about 5' to 3' this way, and 3' to 5' this way. This will be incredibly important when it comes to talking about DNA replication later on in this unit. And one way we often think about the structure of DNA is a ladder. Fairly simplistic, but it does work. So the sugars and the phosphate join together to make the side of the ladder. So that sugar-phosphate backbone is the outside of the ladder, and our bases that go and join up in the middle would then be considered the rungs. However, again, as we know from previous lessons, the ladder gets twisted. That then forms a double helix. So DNA is a double-stranded double helix with antiparallel strands. So that's a bit of a combination there for you with existing information and new information. The antiparallel particularly being the new information for this year. So that's just adding an extra layer then of information to what you previously know. about the double helix. So the discovery of the structure of DNA is actually clouded in quite a bit of controversy. Watson and Crick on the left are the two people who are attributed with the discovery. However, it did come on the back of a huge amount of scientific work, and particularly Rosalind Franklin on the right. Her work in X-ray crystallography really helped Watson and Crick put together a lot of their findings, and it could be said that James Watson actually stole some of Rosalind Franklin's work to make sure that they came out first with this discovery. The model that they're actually showing on the left-hand picture is now actually on display in the Science Museum in London. So that's a picture I took in the middle of their model, which is on display down in London. So in summary of what we've done so far, some of which is existing information, some of which is new information. DNA is comprised of two polynucleotide chains. So poly meaning many nucleotides. Nucleotides consist of sugar phosphate base. Nucleotides bond to form a sugar phosphate backbone. This new bit of information that it runs antiparallel with a deoxyribose sugar at the 3' end and a phosphate group at the 5' end. Those nucleic acids are paired by hydrogen bonding in the center to form a double helix, and those base pairings are specific, with adenine pairing with thymine, cytosine pairing with guanine. So that's what we've done so far. So next up, we're going to start looking at the organization of DNA. So at the moment, we should know that DNA is the genetic material of all living things. We should know about the DNA structure. Heck, we've just done it. We should also think about knowing the difference between prokaryote and eukaryote. Now, some of you might just be going, what the heck? So let's give you a bit. So this little GIF should help us out. Both have a cell membrane, both have cytoplasm, both have DNA, but the prokaryote doesn't have a nucleus or membrane-bound organelles. So pro rhymes with no. No particularly being for no membrane-bound organelles, which includes a nucleus. So can you think of any organism that you've looked at in National 5 biology that doesn't have a nucleus? So prokaryotes, organisms that don't have a nucleus, you've looked at those in National 5. Hopefully, again, you're all shouting at the screen, bacteria. So bacteria are the prokaryotes. Look at all our other organisms that we will be looking at will be considered eukaryotic or eukaryotes. In the coming slides, I'm going to talk you through the organization of DNA in a variety of different organisms and organelles. So you're going to learn some particular information about how DNA is organized in organisms or organelles. So one of the tasks you're going to do today is complete this little table. You can do that as you go, or obviously you can stop later on and come back to it.
So I'm going to talk you through the DNA organisation within different organisms and organelles, and we'll start with prokaryotes. As we discussed, they don't contain a nucleus, so their DNA is in the middle of the cytoplasm, and that's a single circular double-stranded chromosome. Some bacteria, not all, contain plasmid as well, which again are circular bits of DNA. They carry non-essential genes. That's really important when it comes to processes like genetic engineering, which we'll come across later within the unit, and also antibiotic resistance. We'll also see how that information can be carried from one bacteria to the other.
Yeast also contains circular plasmids, and this is an example of actually a genetically engineered circular plasmid that is being used in genetic modification of yeast cells. Next up is mitochondria and chloroplasts. Now this might be something that's quite surprising, but mitochondrial DNA is found in the mitochondria of both plants and animals. Obviously chloroplast DNA is found only in plants. These are both inherited from the mother along with other cell organelles during cell division. Now that comes back to what's known as Endosymbion Theory. It is thought that in the evolution of life on Earth, that large eukaryotic organisms actually have engulfed or brought in these small prokaryotes. So that is why it is thought that mitochondria and chloroplasts contain DNA, because they actually originate from bacteria or other prokaryotes, cells being engulfed by us, which has actually given us, or life on Earth, more capabilities through this symbiotic relationship. So mitochondrial DNA, it is circular and double-stranded. It can vary in size. So those numbers there, BP, those are known as base pairs. So it can go from about 15,500 base pairs to 80,000 base pairs in yeast, up to 2 million in some plants. It codes for transfer RNA, tRNA, ribosomal RNA, and some proteins in the mitochondria, but we'll talk about that more later on. Similar with chloroplasts, it is also circular, it is also double-stranded, and again it can range between 80,000 and 600,000 base pairs in size, and it can also have multiple copies. Those base pair numbers are not something you need to know. This is just a bit of extra information for you just now. And again, it codes for things like RNA, proteins required for transcription, translation, and photosynthesis. But again, more about that later. So irrespective of the base pair numbers and what it codes for, the key bit of information here for your table is that mitochondria and chloroplasts, they have circular chromosomes. That's how it's organized in mitochondria and chloroplasts. It's circular chromosomes. So a key example of a eukaryote is you. The majority of life on Earth is eukaryotic. They contain their DNA in a nucleus, which is in linear form, so linear chromosomes. They also have extra DNA, which we've just discussed. That is in the mitochondria and the.
So here we have a summary of the key bits of information for organisation of DNA in different organisms and organelles that we've just discussed.
So now we're going to look at how it's packaged in eukaryotes. And to give you some numbers to get you thinking, one human chromosome, if pulled out, is approximately 4 centimeters. It packages it into a bundle about 1.2 to 2 micrometers long. That's a millionth of a meter. Again, you have 46 chromosomes. This means you have approximately 1.84 meters of DNA in every cell of your body. So if I just took out the DNA from one of my cells in my body, it would be as tall as me if I stretched it out. But that means if you took every single bit of DNA in every single one of your cells, it's enough to stretch to the moon and back. So how on earth does that get into your cells? So how do you fit all of the DNA into chromosomes and into the nucleus of every single one of your cells in your body? You can almost think about it if you grab a bit of string, hold it in each hand at the ends, and then twist. If you were to do that, or even if you do it, what you'll find is that that string will coil up onto itself. Okay, and that's what happens with DNA, a process known as supercoiling. The one problem with that, however, if you did it with a bit of string, is that coiling happens in a very haphazard manner. Now, DNA needs to be unraveled for producing proteins on a regular basis. If your cells are going to duplicate, that DNA needs to uncoil for replication. So that haphazard manner that happens if you just twist around a bit of string at each end, that supercoiling, that's not exactly what we need for DNA. So we need a bit of order, we need a bit of structure, and we're going to talk in a bit more detail about that. So the first stage of this supercoiling is known as nucleosomes. The DNA double helix is wrapped around proteins called histones. That then forms a nucleosome. So DNA and a histone produces a nucleosome. The next stage is then a thick chromatin fiber. So the chain of nucleosomes then fold again to form this thick chromatin fiber. You can start to see this supercoiling idea, a coil upon a coil upon a coil. Then we have looped fibres. Again, this thick chromatin fibre then folds again to form these loop fibres. So we're starting to really build up this thick loop. We then have even more folds that then finally starts to look like a chromosome. So this chromatin then folds again to produce this condensed chromosome material. So a coil upon a coil upon a coil. So this is the full process that we've just discussed. So we're starting off with the DNA double helix, which you're all well familiar with, but these added extra steps in terms of forming the nucleosomes and then folding up on top of each other, and then eventually again getting back to the thing that you are familiar with, the chromosome at the end. So what I'll do is I'll provide this diagram to you electronically. You can print it out or just save it, whichever you prefer to do. Okay, so effectively the supercoiling is taking this double helix and folding it upon itself, providing an extra bit of structure with a histone, but looping and looping and looping again till you get to this chromosome down at the bottom, which you again would be familiar with.
So to summarise the second half of this video lesson, DNA is organised differently in prokaryotes and eukaryotes, with prokaryotes having a single circular chromosome and or a plasmid, eukaryotes having several linear chromosomes which are packaged through supercoiling. That goes through 4 stages of packaging before it gets to its most condensed state, and histone proteins are involved in that packaging. Eukaryotic cells also contain mitochondrial DNA and chloroplast DNA in green plants, which are packaged in circular chromosomes. And that's us for today. So today we've looked at DNA being an antiparallel structure with nucleotides added to each strand at the 3' end. DNA organisation is different in prokaryotes and eukaryotes, and in chloroplasts and mitochondria, and also supercoiling within eukaryotic chromosomes, allowing it to fit into the nucleus. So we'll carry on this process. DNA in the genome continues with DNA replication next. So if you have any problems, questions or queries, get in touch and I'll speak to you soon. Bye.